Hey everybody! Today we will be talking about the diseases of the circulatory system and lymphatic system, and especially we're going to concentrate on Lyme disease, brucellosis, and leptospirosis. First, we will talk about Lyme disease, which is caused by Borrelia burgdorferi and also rarely by Borrelia mayoni in the United States, and then in Europe it is caused by Borrelia afzelii. We're going to concentrate on Borrelia burgdorferi as the causative agent because that is the agent that causes most Lyme disease. It falls under the domain of bacteria, the family Spirochaetaceae, and the genus Borrelia. It is a spirochete that is helical shaped due to lipid bilayer outer membrane. It has a periplasmic space containing peptidoglycan and endoflagella and a second lipid bilayer in a membrane. It is a gram-negative like bacteria, and it's only gram-negative like because it doesn't actually have lipopolysaccharides. It is 10 to 20 micrometers long and 0.3 micrometers wide. It does not have toxins or secretion system, and it was first identified by Swiss entomologist Willy Burgdorfer in 1982. The flagella that I mentioned earlier are plugged into rotary motors in the cell membrane, and those result in the corkscrew-like motions, just like we learned in the lecture a few weeks ago. On the right side, you can see a nice picture under B, where you can see the flagella in the agent and then the outer membrane. Borrelia burgdorferi is transmitted through the deer tick, which is also called black-legged tick, or Ixodes scapularis on the east coast, and when it appears on the west coast, it is called Ixodes pacificus. It is an organism that grows in the digestive tract of the tick, and then it takes around 2 days to reach the salivary glands to be transmitted to the host. So basically, the tick attaches, cuts through the skin, and then starts feeding. And if Borrelia burgdorferi is present in the tick, it will wander from the gut to the salivary glands and then into the host, for example, the human. At the same time, the tick secretes anesthetic-like substances that numbs the area, and therefore the bite is often not felt. Once inside the host, it rapidly travels through the bloodstream, and it does prefer the skin, the nerve tissue, the joint linings, and the conduction system of the heart. It does like machinery for synthesizing nucleotides, amino acids, fatty acids, and enzyme cofactors, which are acquired from the host. It does not transmit transovarily, so the larva feeding is important, and that's where the tick mostly acquires a causative agent during the larval feeding from infected rodents. It does have a varying gene expression in order to adapt to the environment, and that is important so it can switch between the tick and the mammal. For example, the pH in mammal and the tick is different, and the temperature is also different. The spirochetes, like I already said, multiply in the tick gut, and once inside the host has actually different ways to escape the immune system. It can attach to several different proteins of extracellular matrix or by using adhesive proteins, and for example, the adhesive protein that is used inside the tick to adhere to the tick gut is called OSPA, OSPA. Molecules activate the host response, which eventually leads to disease. So normally the host response activates a localized inflammatory pathogenic response used to clear the infection, but if it gets activated over and over, it will lead to the disease and lesion development. And on the right side, the two pictures, you can nicely see how the spirochetes actually attach to the cell. As for patient symptoms, Lyme disease can be put in 3 different stages, whereas stage 1 and 2 are considered early Lyme disease, while stage 3 is light Lyme disease. Stage 1 occurs normally 3 to 30 days after the initial exposure, and it includes the symptoms and the signs include erythema migrans, which is the typical bull's eye rash, which you can see on the right side, as well as fever, myalgias, which is muscle pain, arthralgias, which is joint pain and headache. And stage 1 is local, and the lymphocytes infiltrate the lesion caused by Borrelia burgdorferi. Then stage 2 occur weeks to months later, and it is a hematogenous spread, which means that it affects multiple organs, and the symptoms and signs include malaise, myalgias, arthritis, arthralgias, and neurological and cardiac involvement. And the most common neurological sign is Bell's palsy, which is facial paralysis, and then also the inflammation of spinal nerve roots and chronic meningitis. And then the most common cardiac manifestation is an irregular heart rhythm. A lot of times, people don't necessarily, 25% of patients don't actually get this typical bull's eye rash that you can see on the right side when they first present with arthritis. And arthritis is actually thought to be an autoimmune response, and often at time affects the knees and other big joints, but it doesn't necessarily even affect both knees at the same time. It does affect one knee at a time, but it can switch between the two knees. Then stage 3, it occurs months to years later, and the signs and symptoms include synovium, nervous system and skin. Neurological involvement can be peripheral neuropathy and encephalopathy that shows as memory, mood, and sleep disturbances. And then some of the symptoms can actually persist, which then is considered post-treatment Lyme disease syndrome, or also chronic Lyme disease. And those are the symptoms that, like I said, persist even after Lyme disease is treated. And those symptoms include fatigue, pain, and joint and muscle aches. In order to diagnose Lyme disease, the patient information like signs and symptoms of the disease and also exposure in possible high-risk areas need to be taken in consideration. And then there's 2 tests that need to be done, the ELISA and the Western blot test. And ELISA is a conventional enzyme-linked immunoassay test, which tests for the blood for antibodies, but not directly for the actual causative agent. And then if that test comes back as positive, then the Western blot test is used, and that tests specifically for Borrelia burgdorferi. However, if the Western blot test is negative, then ELISA was actually a false positive, which can happen since the ELISA is a very sensitive test and sometimes shows a positive even though the person does not actually have Lyme disease. But there's also false negatives that can sometimes happen, especially if the infection occurred recently, because the antibodies take some time to develop. The IgM and IgG are the antibodies that would show in case of the disease, and IgM results can actually be disregarded if the patient has been ill for more than 30 days. And normally results take 1 to 2 weeks to come back. Lyme disease is a zoonotic disease that needs to be reported, and around 35,000 cases are reported each year. However, the estimation is actually around 476,000 cases each year in the US, so quite a few more. Children between the ages of 5 and 15 and adults over the age of 50 are considered to be most at risk. And then high-risk areas include Minnesota, Wisconsin, and the Northeast. But there's also been cases in California and the coasts of Oregon and Washington state that have been reported, but also in the Southeast, like Florida, Georgia, North and South Carolina. And cases are rising mainly due to climate change and mild winters and stuff like that. And reported cases don't actually necessarily mean that it is where the disease was acquired. So I can go on vacation to New Jersey, get bit and acquire the disease there, but get diagnosed in Florida, and that's where the case would be reported. Since Borrelia burgdorferi is a bacterium, the treatment and therapy strategies are generally antibiotics, and the dosage and duration of the treatment is different for children and adults. Some patients also use naturopathic treatments like herbal antimicrobials, supplements, and acupuncture. Some of those supplements include B12, coenzyme Q10, chromium, folate, omega-3 fatty acids, just in order to help improve the repair of cells and to boost energy. And the antibiotics used are doxycycline, amoxicillin, cefuroxime, and ceftriaxone, and they're used according to the signs and symptoms. Some of them are used to treat the typical bullseye rash. Some are treated for neurological Lyme disease, some are used for Lyme arthritis, and the dosage is different, and then also the duration is actually different. But it's generally, you need to take at least 10 days antibiotics. Sometimes it can go up to 21 days. And then if a person is allergic to any of these 3 antibiotics, the macrolides azithromycin, clarithromycin, or erythromycin can be used for treatment. The next disease we will look at is brucellosis, which is caused by Brucella spp, which includes Brucella melitensis, Brucella suis, and Brucella abortus. It falls again under the domain bacteria and the family Brucellaceae and the genus Brucella. It is a gram-negative Coccobacilli, which you can see very nicely on the picture on the right side. Its size is 0.6 to 1.5 micrometers by 0.5 to 0.7 micrometers, so it is actually smaller, I think, than the spirochetes from Lyme disease. And it is a non-sporing and non-motile facultative intracellular organism, and it is non-motile due to the lack of capsule or flagella. Its morphology is smooth, and it is aerobic, but sometimes needs added CO2, and it was first identified by David Bruce. This causative agent is transmitted by sheep, goats, cows, or camels, mainly by drinking and eating raw or unpasteurized dairy products, but it is also sometimes transmitted by pigs. It does have different portal entries, for example, orally by ingesting animal products or the contact with contaminated fingers, then the inhalation of aerosols containing bacteria, the aerosols contamination of conjunctiva or through skin abrasions, and the oral way of entry is probably the most common route. For example, Brucella melitensis is mainly found in sheep and goats, so it's those dairy products from those animals are the way for contamination, and is most pathogenic in humans, but technically eradicated since the 1970s in the US. Sometimes cases are, however, happening due to the consumption of unpasteurized dairy products, and Brucella suis is mainly found in pigs and transmitted through the slaughtering process, or also, for example, hunters that are hunting wild hog are at risk to get infected by Brucella suis. And then Brucella abortus is mostly acquired through occupational hazard, like veterinarians. However, its eradication from cattle is almost complete in the US. Some cases are, though, still there in wild bison and elk herds in the United States. Once it is inside the host, it binds to epithelial cell surface receptor that contains sialic acid and sulfated residues. It does get into the cell by endocytosis by macrophages, where it then resides inside special vacuole for 8 to 12 hours after infection, modifies intracellular trafficking, and transforms the vacuole into replicative compartments, first quantitatively reduce gene expression and protein synthesis in anabolic metabolism, and it has an increased amino acid catabolism. Its replication happens in the ER, and then it leaves the cell to infect other cells. And once it leaves the cell, it will travel to bone marrow, lymph nodes, the spleen, and the liver. And on the right side in the picture, you can see very nicely the endocytosis by the macrophage, and then the replication and the leaving of the cell. Patient symptoms for brucellosis include fever, sweats, malaise, anorexia, headache, pain in muscles, joint, and or the back, as well as fatigue. And some of these signs and symptoms can actually present for a prolonged period of time, and some of them might be recurring or never may go away. And some of those recurrent symptoms include recurring fevers, arthritis, swelling of testicle and scrotum area, endocarditis, which is the swelling of the heart, neurological symptoms, which is in up to 5% of all the cases, then chronic fatigue, depression, and the swelling of liver and or the spleen. In order to diagnose brucellosis, the physician needs a blood culture to identify the bacterium, which is sent to the lab, and the lab actually needs to be informed that brucellosis is suspected. However, you can also take a sample from the bone marrow, cerebrospinal fluid, wounds, purulent discharge, or joint fluid. Once you have the sample, a BMAT test is used to test for antibodies, and the BMAT is a Brucella microagglutination test, which detects all 3 species found in humans, and it is a modified version of serum agglutination test, which is a serological test. And you need 2 serum samples. The first needs to be done within the first 7 days of the symptoms onset, and the second sample or test needs to be done 2 to 4 weeks after to check for antibodies, and for it to be positive, it needs a 4-fold or greater rise in antibodies, and it needs to be incubated for 30 days, which is actually quite a long time, and it does need that long because it has a slow growth. Brucellosis is, once again, a zoonotic disease which needs to be reported. It has endemic areas including the Middle East, Central Asia, China, parts of Mexico, and Central and South America. Around 500,000 cases are reported annually, and around 2.4 billion people are actually at risk worldwide. Both sexes and all age groups can be affected, so there's no preference. The prevalence has been increasing because of worldwide tourism and migration. And then most cases in the US happen due to international travel or rarely consuming unpasteurized dairy products, which are normally imported from neighboring countries like Mexico, and around 100 to 200 cases annually are reported in the US. Since it is a bacterium, the treatment for brucellosis is antibiotics, and it is normally a combination of doxycycline and rifampin for the duration of 6 to 8 weeks. The recovery can take a few weeks or even several months. Normally, there's doxycycline that is taken twice a day orally in combination, like I already said, with rifampin. In acute cases, doxycycline is used twice a day orally for 6 weeks, and then in combination with streptomycin intramuscular for 2 to 3 weeks. Acute cases, they are believed to be more effective and have a lower rate of relapse, so the combination of doxycycline and streptomycin have a lower rate of relapse. And children that are infected that are younger than 8 years have a combination of rifampin and TMP SMZ, which is trimethoprim and sulfamethoxazole. That's a really hard word, and that is used for the duration of 6 weeks. The third disease we'll be talking about is leptospirosis, which is caused by Leptospira interrogans. Leptospira interrogans is also a bacterium under the family Leptospiraceae and the genus Leptospira. It is a flexible, spiral-shaped, aerobic, gram-negative spirochete. It has 2 periplasmic flagella, one at each end, which then extends towards the center of the cell but doesn't actually overlap. And you can see that very nicely in the picture on the right side. The top spirochete is the Leptospira, and you can see that there is a gap between the 2 flagella, where in the Borrelia burgdorferi agent you see that the flagella connect, or that it goes through. The Leptospira interrogans lacks glycolipids but has diaminopimelic acid instead of ornithine in its peptidoglycan. Its size is 0.1 micrometers by 8 to 20 micrometers, so it's more in the size of the Borrelia burgdorferi. And the agent does need B1, B12, and long-chain fatty acids for its growth. And it was first discovered by Japanese workers, and then the genus was created by Japanese bacteriologist Hidego Nogoshi in 1917 when he was working in America. Leptospira interrogans is transmitted through urine of infected animals, which can get into water or soil and then enter through the skin or mucus of the host. The infected animals include cattle, pigs, horses, dogs, rodents, and wild animals. And actually, the infected animals can have no symptoms and actually be asymptomatic. Once inside the host, a generalized infection occurs, but no lesion on the infection site, and normally the agent is eliminated from host tissue, except for the brain, the eyes, and the kidneys, and the agent actually multiplies in the convoluted tubes of the kidneys. Its mechanism is unknown since no endotoxin or exotoxin are associated with them. However, it does interfere with the blood flow and does damage to the endothelial lining of the capillaries, and hepatic and renal function is impaired as well, especially in severe cases, which can actually lead to death. And it is thought that the agent attaches the same way as Brucellosis burgdorferi by attaching to several different receptors of the host cell. It does replicate within the murine macrophages and is then released into the extracellular space. A patient can either show symptoms or be asymptomatic. If the patient shows symptoms, those would include high fever, headache, chills, muscle aches, vomiting, jaundice, abdominal pain and diarrhea, and rash, and it can take between 2 days and 4 weeks after contamination before the patient shows signs and symptoms, and then those can last a few days to 3 weeks, and sometimes even longer than that, and if it's untreated, it can take several months to recover. So those symptoms are considered to be the first phase. Sometimes the patient can become sick again, which is considered the second phase, and then that second phase can result in kidney or liver failure or even meningitis. In order to diagnose leptospirosis, serological testing needs to be done, where MAT is used to detect serovirus-specific antibodies, or a solid phase assay to detect IgM antibodies. And MAT is a microscopic agglutination test, which is generally the cornerstone of diagnosing leptospirosis because it allows to test for specific antibodies. However, it is labor intensive and the procedure is complicated as the Leptospira strain needs to be maintained in order to prepare the live antigen. The agent is present in blood for 5 to 7 days, and then Leptospira specific antibodies, mainly IgM, are produced. The IgM is normally persist for months and might not show an active infection, but a recovered one. And there's also IgM ELISA, which can be used, and it is actually widely used. However, it can, as we learned, it is sensitive and it can give a false positive, and the IgM can actually not be detected in the early stages. There's also the PCR test, which is successful in detecting the DNA in serum and urine samples, but the PCR has expensive reagents and needs a large quantity of DNA, and it cannot identify the infecting serovar. This third disease is also a worldwide zoonotic disease, which is more widespread in tropical countries, and urine contaminated soil can stay infected for up to 14 days. Most of the cases happen during summer or fall, and people that partake in recreational activities in contaminated lakes or possible contaminated lakes and rivers are at higher risk, for example, going swimming or kayaking or stuff like that. It does need to be reported to the CDC, and between 2014 and 2018, between 100 to 200 cases were reported annually. Most of them happen in Puerto Rico and Hawaii, and it is thought to be an underdiagnosed and underreported disease, with a worldwide an estimated 1 million cases with about 59,000 deaths yearly. Getting infected can be prevented by avoiding possible contaminated water. If somebody is going kayaking or wading through water, protective clothing should be worn, and pets that might carry the agent can be vaccinated, even though the vaccine is not 100% protective. Leptospira interrogans is, just like the other two, a bacterium, so antibiotics are used as treatment strategy, and they're once again doxycycline, ampicillin or amoxicillin, are used especially early in the infection. It may require intravenous antibiotics if symptoms are more severe, and then penicillin G is used. Severe leptospirosis leads to multi-organ failure, so it needs to be closely monitored, and treatment needs to be adjusted to the signs and the symptoms in each case. But severe leptospirosis can also be fatal in 50% of the time, even though there is ICU support. Mild cases need to maintain their fluid intake, and for the more severe cases, a clinically appropriate diet should be used. So, for example, in the case of renal insufficiency, electrolytes and protein intake need to be restricted. And lastly, my references over the next 4 slides. I hope you enjoyed this presentation and you learned some more about those 3 diseases.